Welcome to our fourth AfriPopGen webinar of the year. And I, for one, am excited that we have Prof. Segun Fatumo and Dr. Tinashe Chikowore, two great champions of genomic medicine in Africa. Dr. Segun Fatumo will be presenting on his exciting work about enabling genomic medicine in Africa. And in the same vein, Dr. Tinashe will talk about polygenic prediction of type 2 diabetes in Africa. Prof. Segun.
So thank you very much for that kind of introduction, and thanks for the invitation to present today. So my name is Segun Fatumo, and today I'm going to be presenting this work. So I'm currently an assistant professor at the MRC UVRI and London School of Hygiene and Tropical Medicine. I also lead the African Computational Genomic Research Group. So today, I and my colleague Tinashe, we're going to be speaking on a talk that we have titled Enabling Genomic Medicine for Type 2 Diabetes in Africa. So I will start with some background information. I will present our work in the last couple of years, and I will relate this to a new approach called a polygenic risk score that is used to identify subgroup of individual, and in our case, African individual with higher burden of type 2 diabetes and other non-communicable disease in Africa. So it is no news that in sub-Saharan Africa, communicable disease such as malaria, tuberculosis, and HIV have long been among the most prominent contributors to disease and burden. So sub-Saharan Africa is the only region of the world in which infectious disease still outnumber chronic disease as a cause of death. And like you know, with a rapid increase in urbanization in Africa, there's now a trend towards unhealthy diets, obesity, and increases in the metabolic risk factor. So unfortunately, a non-communicable disease is now taking over from communicable disease. And today, the focus is going to be on type 2 diabetes. So although the burden of disease in sub-Saharan Africa continues to be dominated by infectious disease, countries in this region are now undergoing a demographic transition, leading to increase in non-communicable disease, like I mentioned previously. So collectively, WHO estimate that NCDs in Africa will rise by 27% over the next 10 years. This is compared to a global rise of 17%. So NCDs in sub-Saharan Africa are posing an increasing challenge for health system, which has today likely focus on tackling infectious disease. So to effectively address this challenging need, our country in sub-Saharan Africa require detailed epidemiology data on NCDs. So just for the purpose of clarity and not to assume that everyone on this call understand what precision medicine is all about, I'm going to start with to give an illustration of what this means. So precision medicine is an approach for disease treatment and prevention that take into account individual variation in their gene, in their environment, and as well as their lifestyle. So this approach to medicine is in contrast to the current standard one-size-fits-all approach in which the disease treatment and prevention strategy are developed for an average person with less consideration for the differences that we all have. So as you can see on this slide, so the big box on the left show the patient population, so indicating that patients do have different genetic composition, and we are all exposed to different environments. So the standard one-size-fits-all approach assumes that all patients are the same. There's also an assumption that all patients have the same genetic composition, and we are all exposed to the same environment. So the treatment A, for example, is administered to everyone. So the implication of this is that such treatment is not effective for many of the patients. So a tailored approach, we group the individual or the patient and administer an appropriate treatment for each group. For example, from this slide, treatment A, for example, could be appropriate for African living in African, treatment B for African living outside Africa because they have different environmental factors. Treatment C could be appropriate for Europeans, treatment D for Asians, and so on and so forth. So therefore, precision medicine is an approach for detecting, treating, and managing disease based on individual variation in their genetic, environment, and lifestyle, the lifestyle factors. So the terms genomic medicine is a key component of precision medicine, focusing primarily on the potential contribution and the usage of individual genotypic information in their clinical care. So this is how I have defined genomic medicine for today. So for the rest of my presentation, I may be using the word genomic medicine interchangeably with the word precision medicine. So if genomic medicine will be effective and benefit all, there is need for representative genomic research has to be undertaken. A review paper that was published in 2016 relates that 96% of participants in genome-wide action study, GWAS, were individuals of European descent. So 7 years down the line, in 2016, the findings also indicate that the proportion of individuals included in GWAS who are not of European descent has increased by 19%. But the much of the increase that you see as a result is as a result of study from other populations like the Asian population and not necessarily from the African population. So that also still show that there's need for more studies to be done in African population. So this study prompted warning that a much broader range of population should be investigated to avoid precision medicine be of benefit to just merely a privileged view. So even up to recently in 2019, about 79% of all GWAS participants are still of the European descent. And I think you know that using primarily white European population to draw broad conclusion on precision medicine could be misleading and also could aggravate health disparity. So as you may have noticed on the slide here, so the fraction of non-European individuals in GWAS has stagnated or declined since late 2014. This suggests that we are not on the track to correct the imbalance. So this is especially very problematic, as previous studies have shown that African studies contribute an outsized number of associations relative to studies of similar size in the European population. So in this presentation, I will show you an example of such study from our genomic study in Uganda and other African populations. And I want also bear in mind that the continental European population represents only 10% of the world population, and all individuals of European descent worldwide only make up about 16% of the global population. So I think there's an urgent need to expand the diversity of population being studied in GWAS for a more complete and effective precision medicine that will work for all. So as you can see on this slide, governments of over dozens countries have invested heavily in establishing a national genomic medicine initiative. You can see from Saudi Arabia, Qatar, Turkey, Brazil, Japan, even Estonia, with population of only 1.3 million people, and all other population here. So just a few months ago, the Prime Minister of the UK also announced about £200 million for whole genome sequencing of 500,000 individuals in the UK to complement what we already have in what is called the UK Biobank. So in 2016, the Chinese government announced a bold initiative to become a global superpower in precision medicine, which is about 9.2 billion US dollars. And these are all showing the indication and the contribution of government to precision medicine. So the Chinese 9.2 billion US dollars precision medicine initiative could see about 100 million whole genome sequence. And if you have been reading a recent paper from the president of the African Society of Human Genetics, also proposing 3 million samples to be whole genome sequence in Africa. So, of course, many people are familiar with the African Genome Project that is called All of Us, which also aims to enroll 1 million participants from the US and should be completed by 2022. So all these country-level initiatives are an important part of the global effort for precision medicine. I know maybe already you'll have noticed that there is no any national initiative from any African country yet. Of course, the well-known and the well-coordinated H3 Africa initiative is funded by the United States NIH and United Kingdom Wellcome Trust, and not any national initiative. So in a precision medicine paper that was published in 2018, with focus specifically on type 2 diabetes, it was evidence that most of the country level initiatives are public-private initiatives, including the very popular UK Biobank initiative, and as well as the United States All of Us project. So the only exception on this slide is the Genomic Aggregation project from Sweden, which is primarily a public initiative. So this gives us some hope that genomic researchers within Africa may want to look beyond government funding, which will not even be available to them. So about 100,000 years ago, the human migrated out of Africa and started to populate the globe. So they carry with them part of the ancestral genetic variation, so implying that all human, or every human being that you see today, have a genetic origin in Africa. So what this means literally is that everyone came out of Africa. So given the central importance of Africa to human origin and disease susceptibility, so there's a clear scientific and public health need to develop large-scale effort examine disease susceptibility in population of African descent. So African population are known to harbor the greatest genetic diversity, emanated from the estimated 2,000 distinct languages that we speak in Africa, and 3,000 different ethnic groups that is speak in Africa. So in order to demonstrate the value of African genomics, so we recently published our paper in Cell in 2019. So this project represents one of the largest collection of GWAS and whole genome sequence data in Africa. So we collected and analyzed 6,400 people from rural Uganda, rural community in Uganda, and we genotyped 5,000 individuals on 2.5 million Illumina array. So we also incorporated data from other parts of Africa, including some data from Nigeria, from Kenya, South Africa, making a total of 14,000 individuals across Africa. So we have GWAS data from different kind of consortium that exists within Africa. So this collaboration was part of an initiative that is called African Partnership for Chronic Disease Research, APCDR, which was an international network of research groups in Africa to facilitate and promote collaboration research of chronic disease across Africa. So we measure a range of non-communicable disease, and as well as communicable disease, including HIV, hepatitis B and C, BMI, liver function, full blood count, HbA1c level, which is an indicator that is commonly used to diagnose diabetes, which is the focus of our presentation today. So we demonstrate from this study the value of African genome resource by creating African unique reference panel using data set from our 2,000 whole genome sequence individual, and also data from the African Genome Variation Project, and as well as data from the 1,000 Genome Project. So as you can see on this slide, you will see on the top right, our unique imputation reference panel outperformed existing imputation panel in terms of accuracy, especially when you input into East and South African population. So this slide shows the list of novel genetic associations that we found to be associated with various cardiometabolic traits and disease in African population. It also shows a list of distinct associations at already known loci. So as you may have already noticed on the last column here, which has the minor allele frequency of the European population, so you will see what is very common there. You will see is 0, very common here. So what it means is that some of these SNPs that we found, they are monomorphic in European population. So, or they are very rare in the European population. So what this means is that this did not exist in the European population. So in a study that involved only 14,000 African individuals, so we discovered 10 novel genetic associations with cardiometabolic traits. So now these genetic associations are only found in Africa and would therefore not have been found even in the largest study involving European individuals. So our study include a genetic variant that causes alpha thalassemia to be associated with HbA1c, an indicator for diagnosing type 2 diabetes. So this genetic variant is present in 22% of African and almost absent in the European population. So this particular variant is thought to have become even more frequent among African population because it can prevent against severe malaria. So our findings suggest that as HbA1c level are often used to diagnose diabetes globally, it is possible that diabetes may have been misdiagnosed in up to 22% of African based on their genetic composition. It is important to emphasize that several studies have shown that African studies contribute an outsized number of genetic association relative to studies of similar size in the European population. So this study of very small size will normally perform that a study of the same size from other population. So our study also leveraged on the lower linkage disequilibrium in African data sets. So our fine mapping analysis resulted into narrow credible interval for most of the traits that we analyzed. So in fact, we were able to map 16 out of 41 distinct loci to a single SNP in the credible interval. We also resolved previously identified association with HbA1c. So recently, we published a paper in eBiomedicine, so where we describe a number of genomic research initiatives within African population. So in this review paper, we show that the current level of support and the collaboration of we show the current level of support and collaboration within the African researcher. So, for example, we are very familiar with the Asia Africa Initiative. This is an initiative with several studies across Africa, which is aiming to recruit up to 99,000 participants. So there's also a public-private initiative from Nigeria. It is called the Nigerian Non-Communicable Disease Genetic Heritable Study, NCDGHS, which is currently recruiting up to 100,000 participants in Nigeria. And that's the reason why I'm in Lagos today. So we also know about the African Partnership of Chronic Disease Research, which I mentioned before. APCDR has more than 14,000 participants in the network. And also, there are also a few other collaboration and consortia within Africa that are very crucial for a study for precision medicine. So from this key initiative from Africa, what we have seen is high prevalence of type 2 diabetes and a very low awareness of this disease. So, for example, in the pilot study, in our study, the Ugandan General Population Cohort, GPC, so we found that more than 57% of the research participants who have an elevated HbA1c that is more than 6%, which means that they are potentially diabetic, they were not even aware of their diabetes status. And also about 80% of those with hypertension were not aware that they had hypertension. And again, none of those with high cholesterol was aware in our population. So this information are relevant for implementation of genomic medicine for type 2 diabetes and other non-communicable disease in Africa. So this slide shows two important genomics of type 2 diabetes in continental Africa. So the study identified a novel association at a gene called AGMO, which is very distinct from what has been previously known to be associated with type 2 diabetes. So the authors also identified a novel and previously unreported gene called ZRANB3, which may influence susceptibility to type 2 diabetes. But this is also only found in the African population. So this also found there's also an evidence for ancestral specific signal that is distinct between European and African population. So this again further demonstrates why it is important to study whole human population. So we're now using all this information relating to genomics of type 2 diabetes in Africa from the continent of Africa to evaluate the predictivity of polygenic risk score that are derived from European population, multi-ethnics, collectively multi-ethnics, African America for type 2 diabetes in continental Africa, which will be the point that Tinashe will be speaking about in his own presentation. So in conclusion, we know that there's now a global push to implement genomic medicine as a crucial part of precision medicine. But the African ancestral population is still lagging behind, especially the continental Africa. So our findings from relatively a very small data set of about 14,000, we show a great potential to inform genomic medicine. And now there's now need for readiness by African scientists to now take a lead in analyzing their own data. So our findings again highlight the importance of diverse ancestry for uncovering novel biology. So there's a need for large-scale African meta-analysis that can give us even further insight into the genetic architecture of type 2 diabetes in Africa or in continental Africa. So I would like to thank all members of our group that are making this contribution. That's the African Computational Genomics Research Group. I'd like to thank our research, our participants, our partner across the globe from South Africa and other places, Manchester and Imperial College and other places. Particularly my colleague and friend Tinashe, Professor Aisha Motola, Charles Rutimin, Dr. Adebowale Adeyewan, and others. So I'd like to make a special thanks to all the research participants who have given us the access to their sample to be able to do the study that we are doing. And also to the funders, particularly I've mentioned Wellcome Trust, who is funding our study. So thank you very much, everybody, for listening.
Hey, perfect. Thanks so much, Segun, for setting the scene. My job is kind of easy, just diving into us actually seeing how close are we, to enabling genomic medicine for type 2 diabetes in Africa, particularly in the arena of polygenic risk scores. So thanks so much, David, for the introduction and the welcome, and thanks for everyone who has spent time to be part of this webinar. So I'll go straight into it. So type 2 diabetes is at an alarming state currently in Africa, and this situation is not showing any signs of improving. Actually, the International Diabetes Federation is projecting that by 2045, if we're looking at figures in 2019, and then we do projections up to 2045, there's going to be a prevalence of 247% of type 2 diabetes in Africa. And there's been alluded by Segun, you actually see Africa as the highest proportion of people that are not diagnosed for these diseases. So they just are diabetic, but they don't even know. So there are urgent needs and strategies that are required to enhance screening, especially early identification. So with just putting a genetic lens now, we'd realize that the aspect of polygenic risk scores actually show a promise to be such a tool that can enhance early prediction. And there was a study which really started to rock the scene with regards to polygenic risk scores. That was done by Seth Catherson. You can see him there in the picture. And many people started to comment about the impact of this study. So some other people actually say that there's a new test which predicts the risk of serious disease months before symptoms appear. The other one also wrote and said a Harvard scientist thinks he has a gene test for heart attack risk and he wants to give it away free. The others were writing and say clues to your health are hidden at 6.6 million spots in your DNA. So what had happened, Seth Catherson had published together with this group a study that had actually shown that complex diseases like coronary artery heart disease, you could actually develop polygenic risk scores which were so predictive, just like monogenic traits. And this polygenic risk score actually had 6.6 million SNPs in it. And everyone was so amazed. And it did not only end in our communication scientists or newspapers writing about it. Reviews are also written by leaders in the field. We see Segun writing there that we are at the cusp of predicting disease risk for some disorders by means of polygenic risk scores. And we have seen even massive amounts of funding that the NIH is actually set aside for to ensure that polygenic risk scores are well explored. But while everyone was ululating, everyone was celebrating, what needs to be noted or put into context is this study had only been done in Europeans. So later studies that came along later on realized that this story is just good only for the Europeans, but very, very bad for people in Africa. So like Segun has indicated, this is mainly due to the low, actually the very low representation of Africans in the current genome-wide association studies, which are used, I mean, as we're going to see in the methodology, in us deriving this polygenic risk scores. And I mean, Alisa Martin writes in this also paper that also brought this much to the awareness that regardless of the methods that you use, the issue of representation is key. And she actually says that if we're not so careful, I mean, this can actually exacerbate health disparities. But the question then comes to mind, so how much, what has been done in Africa currently? So you find there's a study that was published by, so it was a study that was led by Charles Rutimi and Aisha Motala. So this study actually amalgamated the South African Zulu type 2 diabetes cohort and actually the ADAM study. So then they amalgamated these studies in a meta-analysis, and they did the GWAS, which is the only one, I mean, the biggest that we have in Africans. And you can ask me how many individuals are in this study. They're just less than 5,000. And also in some earlier works that I was involved in, we even started before this GWAS was done. We even took some few SNPs that were known there in 2016 and derived some polygenic, some genetic risk scores, and we realized that no, actually the ones that are significant in Africans predict better compared to the ones that are in the Europeans, something which was later on, I mean, reiterated by studies and low-power studies that were done by Alisa Meta. So the greatest disheartening take-home message is that those European ones, those good news that we're seeing from the European, these PRS predict 4.9-fold less in Africans, and due just to the very low representation of Africans in these studies. So recently also, there was a large, I mean, the Million Veterans Program went on a drive, I mean, to try to do something about diversity. And in this cohort, which is about 1.4 million participants, so this is the biggest study, diabetes study, type 2 diabetes study, GWAS, that has been done of late. And in this study, they increased the proportions of Africans. And as we are going to see, they have about 56,000 Africans in this cohort. But nonetheless, we cannot deny that the Europeans are a million plus. So there were now concerns, I mean, questions that were coming to mind that, yes, everyone is speaking about European-derived polygenic risk scores predicting poorly in Africans. What about the multi-ethnic? So there are some studies. This is a study that was done in the UK Biobank, whereby they actually got diverse GWAS or a multi-ethnic GWAS, created polygenic risk score from it, and they saw it predicted better compared to the European one. And they started writing and saying, oh, this multi-ethnic polygenic risk score improved prediction in diverse population. So at this juncture, it almost seems like this multi-ethnic GWAS might be an answer, but yet this had never been tested in actual Africans, like in continental Africans. They can only be tested in European Africans in the diasporas, the Africans in the UK Biobank, which are actually admixed. So there are some current concerns there that how does these multi-ethnic PRS perform in continental Africans, and do they really predict better compared to African Americans? Because no one has used, I mean, African American derived PRS and compared them with the multi-ethnic or even the European. So this became the aim or the main research question that we're trying to answer in our polygenic risk score paper. So what we end up doing, if you look maybe just for others who might not be familiar with how polygenic risk scores are derived. So you see what you require first, you required a GWAS study. So this is a study that has been done to check associations of a number of variants in millions with a particular trait. And from this study, you get effect sizes of each and every SNP in that. Then after that, you also need a target study. So this is in GWAS, so it's a discovery, the best data. Then you also need another data set which only have genotypic data and phenotypic data. So you're going to combine these two data sets using tools. So in our case, we use the tool called Precise. So how it calculates the PRS, it adjusts it. So it does two things. So it actually gets a number of p-value thresholds. So it breaks down, partitions these SNPs into a number of p-value thresholds. Then within those, it does LD clamping. So you are the one who's set to say, I want to clamp at RR2, 0.5, 0.1. You can play around with those parameters until you get the best PRS. Then after that, you now actually associate. So you take your PRS, which is a continuous variable, and you test it with your outcomes, adjusting for ancestry and also the requisite covariates. So you do this in the target. But in essence, for us to actually know that this works, you also need an additional data set, which is a validation data set, where you actually test it independent of your target data set and best data set. So in our case, this is exactly how we went about it. So we took the Europeans from the Million Veterans Program. As you can see, there are over 1 million. We also had our African Americans, just around 55,000. Then together, the multi-ancestry, which is 1.4 million, as we have seen. Then we tested this. So our target data set was the Zulu study, as you see there. Then the replication, or the one which is independent from the development. So meaning developing the PRS, we use the MVP and the South African Zulu study. But actually to validate it, to check how it works, we actually used an independent study, which was the Adam study, with those numbers indicated there. But so what did we find? So what we saw is actually that the African American derived PRS, in terms of sensitivity and specificity, had actually the area under the curve was higher, was the highest, around 58.6 in the South African Zulu. And when we checked also in the Adam, we realized it was 58.8. So the European, the predictivity was 56.2 and 56.5 in the multi-ethnic. There wasn't much difference. So we then went actually to see if we take this African-American derived PRS and we just add the PCs. So that's that, as we see 58.6. What about if we add age, sex, and BMI? We see it increases, which corroborates with what had been written in the earlier reviews, that if we combine our polygenic risk scores with other conventional risk factors or epidemiological risk factors, we might enhance, our predictivity of the complex traits such as type 2 diabetes. So then there was a question, because what happens when Precise is computing these p-value thresholds, you can see the best p-value threshold for the multi-ethnic was 3 times 10 to the power minus 4. For the African American, it was 5 times 10 to the power minus 8. For the European, it was 0.406. So there was a question that maybe let's really match apples for apples and really see what really is happening there. So what we then did is we tweaked the p-values to make sure we have matched them. So we saw that the African American predicts best at 5 times 10 to the power minus 8. So what about everything at 5 times 10 to the power minus 8? So we see using also the R-squared, the amount of variance that you can get when you're using logistic regression models. So therefore, you see, we realize that the percentage is quite low. So your variance is 0.03 in the European when it's 5 times 10 to the power minus 8. But when you go to the best p-value threshold, it is more than double. Then if you look at the multi-ethnic, actually at 5 times 10 to the power, it's 0.07. Then it increases when it goes also to the best. But nonetheless, all this are less than the African American. And as you can see, even on the bar plots, you can see that just using genome-wide significant hits, when you are using an European, so that's an example of the European, it doesn't predict better. You actually need to lower your p-value threshold, as what we can see here at 0.406, which was the best p-value threshold. So in this sense, we're satisfied that indeed what Precise is selected as the best is really the best, p-value thresholds. And nonetheless, they are still lower, and the African-American predicts better. So the next question was, so fine, this PRS predicts better. So how much does it enhance risk stratification? So if you compare it with the European, so you see it, we just plotted to check if this, because remember, we know there's great genetic diversity in Africa. So the first African is in the Zulu. Then the green one is the West African replication is actually in the Adam study to see how close do they tie. I mean, because of this great genetic diversity, are they going to be different? So we see they're somewhat close to each other across all the decile, save for decile 7, but nonetheless they all are almost similar to indicate that the participants in the upper decile, the 10th decile, have a 3-fold risk of developing type 2 diabetes compared to those ones in the first decile. And actually people in this higher decile, like the upper decile, were diagnosed of type 2 diabetes 2.6 years earlier compared to those in the first decile. So remember the aspect of early identification. Then we also looked, when you compare the cases and controls, like how many people, what proportion, what's the distribution, what's the proportion of people who are in the higher deciles who are in the cases. So we actually see that more people in the cases are actually in the higher deciles compared to the control. So that's just trying to check how much this PRS transforms. How can we utilize it for translation purposes? So what are the key implications that we're getting here? So we see that they still need to increase representation of Africans in GWAS. So fine, there's that paper which had indicated that a multi-ethnic would work well, but if you check in the numbers, like 1,050,000, it's just like a drop in the ocean. So even when we meta-analyze and we aggregate, there's still going to be that Eurocentric bias. And nonetheless, I mean, what's more worrying is at least Africans are at 56,000. But in Africa, actually, it's too generous to say we're around 10,000. We're actually less than 5,000. So we still have a long way to reach to that million. And what's worrying is you see in the MVP, the prediction when they compared the upper decile and the lower decile, it was 5 times. But us, we're still at 3.5. So we still anticipate that, fine, we've seen the African Americans predict better compared to the multi-ethnic. But I think if we increase our own continental Africans, maybe that prediction can actually be better. And maybe who knows? I mean, since Africa is the cradle of humankind, this might even be predictive better in other populations aside from Africans. And there's still this quest to say, maybe we just use one method. What if we use LD-PRED and other tools? How will it all go? So these are still implications and pending questions that we still have. Nonetheless, as based on our study, we could actually conclude that the African-American derived PRS predicts better in continental Africans. So just want to acknowledge and to thank my colleague, Dr. Segun Fatumo, the postdocs and students in his research group, Adam Study, the UGC cohort, the Wellcome Trust, Sydney Brenner. And if you want more information, you can actually go on bioRxiv and actually read more regarding this paper, which is still under review. Thank you so much. Thank you.
So thank you, Tinashe and Segun. I am Noemi. I'm going to chair the question and answer session. So thanks, David, for this opportunity. And so thanks for your presentation and also for like the energy and the determination that you have in pushing your work. I was like really influenced by your energy, really. And so I'm just asking to the audience if some of you have some questions, if you want to just ask some question or write the question. Yes, I saw an hand, right? Yeah, you can say your question. Bigba, if I say your name right. Sorry if I.
Yes, that's my name.
Okay.
I hope you can hear me.
Yeah, we can.
Yes, my first question goes to the first presenter, Professor Segun. He said in the study he recruited about 14,000 what Africans were added, and then they've identified about 10 novel variants. What's it? So I want to find out the inclusion criteria, because when we talk about recruiting Africa, we also have people in Africa who are actually not Africans. And it's always been a challenge if you are looking for the true nationality of a person, if you want to include to a study like the GWAS that we are talking about. I want to know whether maybe there's a model they have used to be able to know who was a real African that they included in the study. Thanks.
So thank you very much, Big Bright, for your question. That's a very good question. So I will try to answer it this way. The process of doing GWAS, there is a need for doing quality control. But even before getting to that stage, so our aim, we went to the rural community in Uganda, and we recruited 5,000 people that were genotyped on the Illumina array. We also recruited an additional 2,000 people that were whole genome sequence. So that's our cohort in Uganda. So we also collaborated with a study in South Africa, who also recruited people from South Africa with a very clear criteria. And also the Adam study also recruited people from Nigeria, Kenya, and other places. But what I want to say is that in our study, in GWAS study, we have a process. The first process is a quality control step. And quality control step is a step that is very key to be able to, we can use a method called PCA to be able to put aside where people are from, to know what ethnic group are they from Africa. And the people that do not relate to what we want can be discarded. Apart from your question also, we also went as far as saying maybe there's possibility to have one person be recruited like 2 times in the study. That kind of thing also could create bias in our study. So those kind of issue where those kind of individuals were also discarded from our study. You may also know that in African population, people that are very related, they tend to live together in the same community, especially in the village. And this also could create bias in our study. So we also adjusted for that. In case we have people that are very related, they have a very similar genetic information. So we also adjusted for that using appropriate method. Thank you.
Right. Thanks very much. Am I allowed to ask the next question?
Yeah. It's if you have another question, yeah, it's fine. Then we will go to Jorge. Yeah.
All right. My next question is about the second presenter. Polygenic risk score, if you look at the association and the impact, is a bit small. For instance, you may have about less than 20% association with the phenotype. Like most of the studies I have read in terms of polygenic risk score, if you are looking at it in terms of clinical interpretation, the evidence is not all that enough. That's what I've heard. I don't know if he has also come across that study, and is it because maybe the data is not enough, or I don't know. Maybe if you could help me out here.
Okay, sorry, I didn't really get. Can you come back again? Just reiterate your question. Sorry, I just want to understand the context. Right?
Yes, please. I can hear you.
Yes. Can you reiterate your question?
I was asking of if you look at polygenic risk score, if you are interpreting the results in terms of like clinical predictions and stuff, you would realize that the impact of the collective, let's say, association of the polygenic risk score to the phenotype is not all that much. Yes. Like for instance, yes. So I don't know how. Is it because maybe the data may not be enough, or if you have enough data, that is how polygenic risk score interpretation is based on?
Yes. So you're correct there. I mean, the polygenic risk score on its own doesn't predict much. So that's why most reviews, like how I started to say, people are saying a PRIS, a polygenic, in conjunction with other epidemiological markers can actually make us get a better scoring at the end. And even if you see in the raw curves that I showed there, you can see if you just add your PRIS and the PCs, it's like 0.56. It's not much. But the moment you add BMI, you add age, it shoots up to 80. So it's not an issue of sample size. I mean, this has also been seen in Europeans where they have hundreds of thousands and millions. So it's something that's there. Fine, there's still quest to say, I mean, let's come up with better methods. I mean, we know NIH is interested in that, to say maybe it's just the methods that we're using, but it's not really an issue of sample size, because they're saying as low, you need like a target sample as low as 100. I mean, so most studies have more than 100 as a target. and you need indeed a larger discovery. The larger your discovery data sets, the more your effect sizes actually get closer to what might be seen regularly. So it's not an issue of sample size, but that's how it is. I mean, and this is also, remember, PRS did not start to boom now. It started before. So this is just a rejuvenation. I mean, when Seth Kastersen then showed that, no, look, this thing can even predict close to just like monogenic traits. But before it had started, around 2006, later, even around 2014, people were publishing. Then it went down. People were saying, these things, I mean, they don't do much. But now, so the gist here, it's not really, it's just trying to enhance stratification. So if you can put people in deciles and you see someone in the upper decile, I mean, was developed diabetes 3 years earlier. Or if you are using a European one, so they only say you have a 5-fold risk, but they haven't translated it to say in terms of years, then it can something that can be applied. And you can even stratify people according to those and tease out further interesting questions. So it's not really the a sample size issue, but how the understanding is currently.
Just to add to that, when you are doing PRS, you are not, for example, if you have a population, you cannot predict that everybody will have type 2 diabetes in that population. You are everybody is familiar with the normal distribution. It is most likely maybe about 10% of the population or less or more we have diabetes or the particular disease of interest. So what PRS is going to do is to be able to, how can we know this 10% or this 20% that have PRS in the next 5 years or 10 years? And that's what it's going to do, because not everybody is going to be disposed to have that particular disease. So we want to identify those subgroup of individuals that have the high burden of the disease.
Yeah.
Thank you very much. I am satisfied. Thank you.
Jorge, do you want to ask your question?
Hi there, guys. Thank you for some excellent talks. I really, really enjoyed them. I love the precision medicine work going on in Africa. My question is a bit open and it's technology related. Accounting for the high linkage disequilibrium in Africa and the incredible diversity of the peoples there, do you think we'll see a shift of moving away from genome chips to capture data to whole genome sequencing methods and then apply scores to those using those technologies, or not? Just opinions. Anything's fine.
I think that's a very, very brilliant question. And the way I will answer it is, it's still a function of fund and kind of funding that is available. I know that the cost for doing whole genome sequencing is now coming down, but if you compare the cost of doing whole genome sequencing with doing genotyping, it's still very, very different. And I'll give an example. We are currently working on doing genotyping of another 6,000 individuals from Malawi, and that will cost us about £500,000 for doing genotyping for 6,000 people. And that one individual will cost about maybe $60 thereabouts to do genotyping for one person. But even though the cost of doing whole genome sequencing is coming down, but for one person for whole genome sequencing is still about $600 to $1,000. That's very expensive compared to doing genotyping. So I think it's more, we're going to get more information from whole genome sequencing, but it's still very expensive and funding might not even allow for that.
Great. Great answers. Thank you.
Tinashe, do you want to add something on this question, or should I? Okay, so I should move on to Chris.
Hi there, a question to both speakers, I guess. And I think most people on this call would heartily agree with the idea that there should be a massive expansion in GWAS activities in continental Africa. The really boring but serious problem is money, because most African countries, even middle-income countries, don't have the kind of research grants that can fund large-scale GWAS studies. And obviously there are initiatives like H3 Africa, etc. But I was wondering to what extent you think that non-African countries, say Europe, America, etc., are prepared to invest heavily in a large expansion of GWAS studies because of the recruitment and the genotyping, etc., is still a really big target and big grants.
So, Chris, that's a very good question, and the answer might be controversial. So, I think I'm one of the people that will look forward to the government in Africa spending their money for research in Africa. So, I think I don't agree to the fact that we must rely every time on the United States NIH and the United Kingdom Wellcome Trust to fund our research in Africa. So I think the government in Nigeria, the government in South Africa, the government in Ethiopia and other places in Africa should look into funding research in Africa instead of us relying on the other continent to fund the research. That's the way I will answer it. And you're already aware that in the last 8 years, we asked a lot of funding for East Africa. But those funding has come mainly from other places, has come from NIH and Wellcome Trust. I think this is time for the government in Africa or NGO or other charity organizations to fund research in Africa.
Yeah, maybe also to, Chris, I think this is also the time, fine, maybe our governments might be castrated with many priorities, but also I think this is where we need industry government partnerships, whereby I think also our big tech companies or genetic companies should also see the potential or value of genetics, as like what Segun you're doing in Nigeria there, whereby it's a private company that is partnering and they are trying to recruit 100,000. That is even bigger than what Wellcome and the NIH have done through HT Africa. So I think there's need to be that broad scale. Yes, we know Wellcome Trust is also trying to establish a bigger African cohort. We're not so sure whether they're going to add genomics to that, but they have a big call whereby they want a very large African cohort. But I think we just need to have many. I think we should just look. I think many players should just jump to the boat, and maybe also for us as researchers. We need to show. I think if we can also show that Africans predict better in other Africans or the value. I think it's for us, for researchers, to show the value, and there's great work happening. For instance, the landmark paper that was done, by Sean and others, and Anand, Sean is on the call. Just showing that, there's still a lot to be explored from African whole genomes. I think such things will entice many people to have interest, and even the great story of PCSK9, that has led to a drug, which is a blockbuster drug that lowers cholesterol by 60%. But that was just found in people of African ancestry who just had these variants, and someone could translate it, and now boom, we have a drug. So I think, fine, is we want funding. Also as scientists, let's make it much rosier, such that even if people don't want to inject money, they see, wow, there is gold. And yeah, I think it can change the trajectory. But I feel we're getting there, maybe, but the pace is quite slow, very slow. Yeah.
Thank you very much, Tinashe, and great answers. Thank you.
So I will read a question here in the chat box. So thank you so much for the insightful presentation. Is the UG2R up for us for imputation for researchers doing GWAS on African samples? How is the data accessed? They ask.
Okay, so we have the Uganda Genome Resource available for access. So after we publish our paper, so the data set is now available to anybody. Can reach out to me for data access, and they should be ready to fill out the necessary data agreement form, and that can be shared with them.
So I think we are perfectly on time. Maybe I can just ask a question for you. Like, I just have a curiosity and a question about polygenic risk score in Africa. I would like to ask you, how would you implement a polygenic risk score taking into account of all the different subgroups and individuals that are present in Africa? Like, if you think more about some, I would say more national, for example, polygenic risk score studies, or more a kind of more continental, because I think that could be a huge genetic difference among all the continent. So if you thought about a kind of project on this.
Yeah. So there is, I mean, not to spill the beans early, but there is a potential of such a project actually starting maybe around June, July. I mean, if all goes well, where there are many people are harmonizing, I mean, genomic data sets across Africa to tease out all these questions. But what we can say from other papers that we're working on, aside from type 2 diabetes, we see that predictivity varies. For instance, if you cluster people in Southern Africa, Eastern Africa, Western Africa, for instance, when you're looking at lipid traits, you see actually the prediction is different. When you take BMI, we have such papers in the pipeline, we can see the prediction varies. But somewhat different to type 2 diabetes. You see, the prediction is almost the same. So I see this is going to be a trait by trait, because the whole issue, it all depends. The variants that are in that polygenic risk scores, do they vary in effect sizes across, or do they have different allele frequencies in these ways? But if they are almost the same and they are so stable, because we cannot assume to say all variants are going to be very different, or the L dependents are going to be, it's just going to depend with that. But like what we have seen in our type 2 diabetes, which was quite amazing, that, I mean, because Adam is a mixture of Eastern Africa and others, but still, like if you take something from Southern Africa and test it also in other, it's actually the same, almost the same. So I think we'll need more studies trait-wise. Then also, I think it will be very much interesting to explore this region-wise, maybe country-wise. I think it all depends. If we have much funding then to do that, because also the representation, like in the paper that Segun is saying, the review, you can see most of this work is clustered too much in South Africa, and also North Africa, then just scattered around there. So I think maybe we'll need more resources to go country-wise. But I think for now, region-wise, it's much more interesting. Just how to see it and maybe tease it out at trait level, because various traits will have different patterns. So I know there's a paper by Alisa Martin where she says it varies, but she looked at particular traits. But for us, for type 2 diabetes, it didn't. But we know for lipids, it does. For BMI, it does. Yeah, thank you.
And just to add a bit to that, I think our PRS will be implemented in Africa and even other places all over the world. It's still very difficult. I'm not even sure whether the UK and the US are very clear yet on how it will be implemented. I think we are still at the elementary stage of PRS, and every one of us all over the world, we are still figuring out how it will be implemented. There are a lot of issues that relate to PRS. There are issues around ethics. There are issues around a lot of stuff and all that. So we are still at the elementary stage. If anybody is interested in doing PRS for their work, this is the time to do it.